Today, as you mentioned, I'm going to be focusing mostly on applications of deep learning module in Visiopharm software, and at the very end, talk about a technique that we're trying to develop in my group to essentially use a vector 3 to do cyclic IF. So as you mentioned, we're in the Traction platform, which is the translational, it's one of the translational arms of the MD Anderson Moonshot programs. And my job as a functional group leader in imaging is to interface with all the different aspects of the Traction platform and to partner and collaborate with other groups outside, within MD Anderson for things like clinical trials. And also work with corporate partners to develop late-stage drugs for them. So we have kind of a combination of different technologies. We have discovery and innovation, understanding aspects of tumor biology. We have translational biology, which is currently where my group is situated, right in the middle here. And we also work with drug development side as well on things like biomarkers. So today I'm going to be highlighting 3 different technologies that we've developed in our group and show how we've applied imaging tools to all of these things. So that's essentially creating custom tissue microarrays, using Visiopharm and deep learning to do image analysis, and also essentially cyclic IF and high dimensional imaging at the very end. Okay, so the examples I'm going to talk about are using a decision forest to characterize the PDX cohort, using deep learning to evaluate drug combinations, and also highlight the different phases of mitosis in some IF data, and lastly, find ways to define rare cells and evaluate targets with cyclic IF. So what is deep learning? Well, you've probably all seen some version of this slide if you've ever seen a lecture in artificial intelligence, but it's a subset of machine learning and a subset of artificial intelligence. So what that means, more specifically, is if AI is thought of as a computer's ability to perform tasks that require human intelligence, like visual perception or decision making, deep learning specifically refers to artificial neural networks. And so a set of algorithms or sets of rules, essentially, that are multiple layers deep that allow a computer, upon certain types of training, to make decisions. So when we talk about a stacked neural network or the deep learning depth, what I mean by that is that essentially what you have is several hidden layers of categories or sets of rules that you've defined or baked into your algorithm. And the number of layers is important for the data recognition process. Another aspect of deep learning is automatic feature extraction. And one of the best examples of this type of thing is in basically autonomously driving cars. They're essentially taking features from a visual picture and turning that into a stop sign or a dotted line down the middle of the road. And lastly, deep learning networks, neural networks, have feature hierarchy. So I'll give you an example of that in just a second, but this is where each layer of nodes basically trains or bases its decisions on the next layer as it goes. So you can go from literally just pixels on a screen to edges and lines, to parts of an edge, to actual object models. And I'll give you an example of that in just one second. So if you were to look at a mountain range right here, and I mean, obviously you want to avoid anything that's not supposed to be there. You only want to find the trees. You want to avoid moose and other wildlife. Then what you can do in this particular case, if you have different size trees and different types of trees, is you can find one tree that you think fits many of those aspects of what is, makes it a tree, or it has the highest treeness capability. And you outline that, okay? You define as they say, the base truth inputs for all of that. And what a deep learning algorithm is going to do is take that and consolidate it into one node layer. And as I mentioned, these are taking things down at the pixel level and acquiring abstract features from that. So edges, contrasts, essentially, you could think of this in this particular example as where a needle, a pine needle is juxtaposed to the sky, or a pine needle is next to another. And then what they do is they turn that into another layer, okay? And so some of these features become a little bit more fleshed out. You have the edge of a branch, you have the bark, etc. And then eventually those, after multiple layers stacked to neural networks, those turn into recognition of complex features like these trees. So just to give a quick example of this in action, essentially, if we have one type of algorithm which is called the decision forest, which is a lot simpler and a lot faster than deep learning algorithm, essentially you can see already, if you compare the very top image to the bottom image, that we've got rougher edges. Now, part of this is because we did this at 2x magnification, and the other part is that essentially a decision forest is really good with categorical data, right? So if I were to take a H&E image like this, and I said, okay, everything that's tumor, I want to put a blue mask over it. Everything that's necrosis, I want to put a red mask over it. And everything that's stroma, I want to put a tan or khaki mask over it. Then what I'm going to need is a really large training set. As I said, in this particular case, I needed at least 20 plus images. But essentially, as long as I annotated those well, it didn't need a lot of assisted learning with this. I needed to clean up. There was a lot of post-processing steps where I essentially had to say, well, if you see this, change here. If you see this, change here. But the good news here is, and this is why I bring it up, is if you just need to get a rough idea of how a tissue microarray is looking or a huge data set is behaving, this is extremely fast to run. The decision forest took seconds to run, probably because it was done at 2x magnification, and it is dealing with highly stratified categorical data. So on the bottom, you can see that the resolution is a lot better. It's really defining those edges. And I made a point when, even though I did do a small training set, what I tried to do was find all the regions where these edges exist and really help the algorithm decide where to make those boundary decisions. This did take basically over a day of sitting there doing iterations over and over and over again, essentially minimizing loss in this particular algorithm. And it did need assisted learning, so I had to stop it a couple times and test the algorithm and then make sure that it wasn't making the wrong assumptions. The nice thing is that it worked at 40x magnification, so it basically was able to see a really high resolution of things, and it required literally no post-processing. However, it was slow to run because it was working at 40x and it had to take a lot of things into account. So as I mentioned, these are all the decisions you kind of have to make when you are doing image processing. It's what your data set size is, what your type of information is. And so the reason I bring up decision forest first is if you have an IF image where it's very, very clear where the boundaries are for a stroma, here we have it labeled with alpha SMA, or tumor cells labeled with HLA, then you can pretty easily create a fast decision forest to quickly give you a rough idea of where all the tumor cells are, where all the stroma cells are, et cetera, and do this for an entire PDX. So I created this PDX of a set of essentially xenograph models that we have. This is all just untreated models, and I made a point to include cores from both the outer part of the tumor and the inner part of the tumor, and also controls for things like immune cells and general tumor cells. But what's nice about using a decision forest is that in a pretty short period of time, you can stratify this data. So one kind of cool thing I was able to do with this, just as an example, is to rank the percent tumor for each of these, right? And I didn't have time today to put that into the inner tumor and outer tumor, but that's definitely something you can do. You can say, okay, what's the percent tumor if the core was from the outer part of the tumor? What's the percent tumor if the core was from the inner part? Well, one kind of nice feature we see already out of this is that when you rank based on the percent tumor, you see that generally anti-correlates with the percent stroma, but it doesn't really correlate with the percent necrosis. And this sort of, the necrosis isn't as informative because these are untreated samples, but nonetheless, it's quite interesting. Okay, so next I kind of wanted to show you an example of if you have, and this is the case in our department, where you have lots and lots of H&E slides, and we're constantly trying to evaluate the biology of different drug combinations. This is something that we assist companies with on a day-to-day basis. And we need to understand if you did treatment 1 and treatment 2 versus treatment 1, 2, 3 versus treatment 2 and 3, how do you start? How do you begin to understand what exactly is happening to the tumor? And because we have these slides, there are lots of trainable features in hematoxylin eosin slides. As you can see here, just kind of similar to what I was showing before, you have the stroma is pretty identifiable. The tumor is fairly identifiable, and I'm sure there are pathologists in the audience, and I'm doing my best on here. I'm not a trained pathologist, so maybe if there are certain people who think that I'm making a mistake here, please let me know. We have necrosis in this region. We have inflammatory cells on this boundary right here, blood vessels and vasculature, red blood cells, and then fibrosis, which looks a little bit like stroma, but doesn't have any nuclei. So based on all of these different traits, we can train an algorithm a deep learning algorithm to go from H&E sections of tumors that look like this. As I said vehicle, treatment 1, 2, 3, etc. And we turn them into this, okay? And there's a wealth of information in all of this. And this is just scratching the surface, because since we used scans of a 40X off of a Polaris on all of these, if we were really careful with the training sets, you could do a cell-by-cell resolution if you were looking for something very, very specific, or you had a tissue type that had a very recognizable cell type in there. And then what we were able to see was that with vehicle and treatment 1 and 2 alone, we didn't see that much variability in the percent total area of tumor, stroma, and necrosis. But when you start looking at the other combinations, really interestingly, we found that treatment 3 alone led to almost the entire tumor taking over everything, with very little stroma and almost no necrosis, which we found very, very interesting from a biology standpoint. Whereas if you did treatments 1, 2, and 3, we see that the tumor is completely just blown apart. There's almost no recognizable tumor tissue available, and a lot of stroma. It turns out that I actually can, if you were to look closer at these, I could probably subdivide the stroma into stroma and fibrosis. So it's actually mostly fibrosis, but for the sake of simplification, it was stroma/fibrosis. And interestingly, a lot of, good amount of necrosis, but not as much as, for instance, treatments 1 and 2. So if we look closer at these, H&E sections, what we find is, just looking at the tumor for that treatment 3 alone, where there was almost no stroma and almost no necrosis, that treatment is creating a highly vascularized tumor, which is why we're not seeing crazy necrosis. However, if you change it to treatment 1, 2, and 3, as I mentioned before, you see that the proliferation of those lumens, so the number of columns of cells for each of those lumens goes down precipitously, and you have an increase in fibrosis. So essentially, we're able to look at these big data sets, and by getting a snapshot here, we can highlight unique qualities of the biology, which will help us inform down into more direct biomarker-based studies. So next, let's say, as I said, if your resolution is really high, you're doing 40x scans, or if you have fluorescent markers, where not only is the specificity extremely high, fluorescent antibodies that help you identify certain marks, but the IF resolution helps you identify different morphologies. You could do something like this next example that I'm going to talk about, which is that if you were to look at phosphohistone H3, which is a marker for mitosis, looking in IF, you can see there are actually 6 different phases of mitosis in here, and they're clearly delineable by their morphology. And this is something that you can train an algorithm to do. And so you can go from this, beautiful stitched, this is actually I believe this is a 20x scan. So we were able to get this kind of information off of a 20x stitch scan on a Polaris. You can go from an image like this, where you can see there's a heterogeneous population of cells here, of phosphohistone H3. And this is, you can see it took a lot of training to do this, 272,000 iterations in a deep learning algorithm. And what you can do is you can tell it to ignore everything else. So non-phosphohistone H3 cells, after the training session, essentially it gives you a heat map that says, I am extremely confident by this red color, indicating by this red color, that I don't need to know anything about all of these different things. All I care about is this. So for background, for non-phosphohistone H3 cells, it's good. I don't have to worry about it. Then if you train them all appropriately, for each of your phases in mitosis, it will identify those, right? So it says with very high confidence, here are all the interphase cells. And you can do that for all these particular things. And in this particular case, there was a category where we just, you can't really define what exactly that was. And so we called it mitotic collapse. We did define what it was, and we think that this is examples of mitotic collapse. So we turned one marker into 7. And when you put a mask over like that, essentially you get something like this, and you can plot this in multiple different ways. We're still figuring out the best ways to do this. But one way is literally just a stacked, kind of a stacked pie chart kind of area. And you can see that with vehicle, treatment 1, and treatment 2 by themselves, we don't see a big change in the variability of the composition of all of these areas in mitosis. But if you add treatment 1 to treatment 2, what you get is a huge increase in the interphase prophase area, which is almost showing that there's a slowing of mitosis. And another way to demonstrate this is using a radar plot, because we know this is a cyclical event that leads from interphase to prophase, prometaphase, metaphase, anaphase, telophase. And what we can already see is in the combo treatment, this upside down triangle here, you can see that it's highest in this particular area here. So it's almost getting stalled in mitosis. Now, what I should say is these are all really kind of fun applications of deep learning, and so far they've been giving us really interesting insights into the biology of tumors that either we don't have time to create multiplex panels for, or we just have so many samples that it's worth it to get a broad overview at first. What I will say though is that at the end of the day, these are, for a lot of these are heterogeneous tumors, right? And so depending on the question that you want to ask, sometimes deep learning just isn't going to answer that question. I mean, there was actually one example I was trying to prepare for this talk, and it was using deep learning to define the epithelial versus mesenchymal nature of a tumor. And I kept comparing it back to the IF, and this particular tumor had a few regions with epithelial areas and then a few regions with mesenchymal areas, but after a treatment, it kind of blended together. We had some sparse cells that were epithelial and ones that were mesenchymal. And what the algorithm wanted to do was kind of make a decision based on that categorical data to say, okay, that's mesenchymal, that's epithelial. And so it wasn't perfect for that particular application. So something that I thought would be nice to highlight here as well, also considering the fact that a lot of people's labs have been closed, is kind of taking reagents that we had in the lab already, like a vector 3, and seeing if we could evaluate targets and define rare cells via cyclic IF. So why would we use cyclic IF in the first place? Well, it's really simple to prepare, simple preparation. It's amenable to tissue microarrays. It's quantifiable signal because it's IF. It has higher cellular detail than imaging mass cytometry, especially if you're imaging at 20x. Even 10x, I feel like, is probably at certain stages similar to IMC. You can do 50 plus potential colors using that, and if you have markers that are fairly well established, it's easy to create and test panels. The con is that, especially the way that we're currently doing it, is that it's time intensive to image all the rounds of staining, and it's definitely time intensive to perform co-registration and analysis. So that is one benefit to, if you were to choose the Codex platform to that, is that it's all kind of integrated in its own thing. So the protocol that we've been using and testing, and this is still in the testing phase, is called the Milan technique, which is multiple iterative labeling by antibody neodeposition. And it was created by the Cataretti lab in Milan. And basically it starts off very similar to essentially any regular IF stain. You have your dewaxing, rehydration, antigen retrieval, and we only do one round of antigen retrieval in this, notably. Then we apply block, we apply our antibodies and DAPI, and cover slip and image on our Polaris at vector. Well, we have a vector 3, there's a Polaris on another part of campus. We do it at 10x because it's fast. We can get a tissue microarray done in about 20 minutes, and then you decover slip, okay? And then essentially you incubate at 56 degrees with SDS and beta-mercaptoethanol, sort of the way that you would strip a Western blot. Then you wash with TBST and start all over again. You apply block. You have to apply block at each of these stages. You apply your antibodies, whether they're conjugated antibodies or whatnot, and you always apply DAPI because we found that SDS and BME does strip DAPI. Cover slip and image that on a Polaris again. And essentially you just keep going for as many rounds as you need. So kind of just as a proof of concept, I wanted to show you an observation we saw with treated and untreated samples. Now this is one particular case where we didn't know anything about this tumor. It was a total pilot study, and I just thought it would be a nice way to turn essentially a study with 20 mice into one tissue microarray and see if we could recapitulate some of the things that we've seen in the staining. So what I'd like to direct your attention to in this is, if you look at phospho-S6 in the untreated tumors, it's more or less more uniformly localized. There's a fair amount of S6, the same amount of S6 on the outside of the tumor as on the inside. And this is sort of true for a hypoxia marker like GLUT1. If you treat with this drug, however, you see a massive increase in phospho-S6 on the inside. And overall, interestingly, you don't necessarily see an increase in GLUT1. If anything, it maybe goes down a little bit. So I guess what I wanted to see was, this is just a pilot. I didn't look at all the different samples, but if we were to create a tissue microarray that includes cores from the inner and the outer parts of the tumor, and we took 4 cores from each sample, each block, and essentially created a tissue microarray that that was when I say created, I mean I created this myself. We didn't have a core do it. All the mice are from vehicle and treated are now on one slide, and they're set up so that we can identify which is which. Then you can set up in this particular case, it was 7 rounds of staining for various markers and image slides, just as I said before, with your 10x objective. Perform the co-registration of those 7 QP-TIFFs straight off the vector 3. Perform cell segmentation on those. This is all in Visiopharm, by the way. And then phenotype those cells by serial thresholding. And what you'll get is essentially an ability to visualize the phenotypes by t-SNE plots, profile plots, etc. So if we were to just look at the raw data, one thing that I've been trying really to do in our group is to figure out ways to get a snapshot of what the data looks like before we go into all this massive deep dive in analysis. So you say, so that when you get to the analysis, you say, does this make sense? And it can be difficult to do that with individual slides, but with tissue microarrays, sometimes you can get a snapshot of what's going on. So in this particular case, it looks like phospho-S6 is definitely being modulated, but it doesn't seem like it's obvious. It's obviously all going down or obviously all going up. There seems to be some interesting modulation there, whereas you have certain like hypoxia or stem cell markers, which look like they're more or less going down. And things like vimentin, which is an epithelial to mesenchymal transition, or it's a mesenchymal marker, my mistake. It looks like it's maybe going up. So what you can do is you can start to take individual cores, and this is just to give you an idea of some of the data that's generated on here between the treated and untreated. And this is something you have to be careful with. I'm de-cover slipping a slide 8 times, but one nice thing you can see is that the tissue is more or less, pretty intact after all these. As long as you're careful with de-cover slipping the slide, we're not treating with crazy heat. We're not doing anything really out of the ordinary other than just treating with SDS and beta-mercaptethanol, and then washing a lot and making sure to block and put DAPI on. Okay, so just to kind of wrap things up, what you can start to do is you can plot the inner cores from the outer cores. So I'll say the reason I did this was because if I grouped everything together, I noticed that the changes were definitely less dramatic than we expected. So kind of recapitulating that IF image that I showed you before, the inner part of the tumor is definitely increasing in phospho-S6, while the outer part looks like it's decreasing. You can tell the data, this is fresh off the press in terms of data generation. So I haven't performed stats on this, but this is definitely indicative or suggestive. And the nice thing is these are 12 or so cores from each of these, so the numbers are pretty good. You see GLUT1 decreases in the inner part of the tumor, whereas it doesn't really change at all because we didn't really see it very much on the outer part of the tumor before. And then you can see that like proliferation markers like this, phosphohistin H3, are decreasing on the whole. So the drug is definitely decreasing proliferation across the entire tumor, but we also see local changes as well. And one thing, just to not to do a crazy further complication of things, is you can start to look at more rare cells. So cells where they have more complex phenotypes. So in this particular case, phospho-S6 and vimentin is much more likely to increase in the inner cores than in the outer cores. You can actually see that if it was more well distributed before that, after this particular treatment, you see that essentially prominence of the S6 and vimentin on the outer part of the tumors is basically gone. It's basically zero. So with that, just since this was more on the technical side, I guess I just will highlight that deep learning is definitely useful in cohort evaluation analysis. So if you have a TMA or you have a whole set of PDXs, especially if your samples are very different, if the morphology of those samples is very different, then your stacked neural network can really help save you a lot of time that would be difficult to do that same thing with a decision forest. One nice thing is that it allows you to utilize existing reagents that you already have, like H&E scans. And as I mentioned and showed you an example, it can be used to evaluate the biology of drug combinations. And lastly, I feel that it already augments existing IF data sets. So for instance, phases of mitosis, and this is something that we're constantly evaluating and just really trying really hard to push the boundaries on the reagents that we already have, right? Well, how do we get more information? How do we get higher quality of information from existing data sets that we already have? As far as cyclic immunofluorescence, the way that we have started doing it is fast and inexpensive, but it is a little bit tedious with respect to analysis. It's useful when deciding on IMC panels, and by that I mean imaging mass cytometry, which is another way to do high dimensional analysis. And that's really what we're using it for, is almost a stepping stone to informing on what direction is an imaging mass cytometry panel going to lean. And then when used with a TMA, it can be helpful to identify tumor subsets that respond differently to the drug. So with that, I really once again like to thank the organizers for this, and basically the Traction department and my boss for allowing me to give this talk today, and open the floor to questions. Thanks very much.
Thank you very much, Dr. Daniel. I appreciate that very thorough presentation. And we do have some questions. Before I ask the questions, I will remind everybody, if you have any questions for Dr. Daniel, please open the Q&A panel and type them in, and we'll get to them. First question came during the earlier part of your presentation. The question was, for treatment 1 and 2 and treatment 3, these tissues are from different patients? Is the question.
These are all preclinical models, so they're all from different mice. Yeah, so they're PDXs that were taken from different mice.
Great, thank you. The next question was, could the deep learning be applied on codex images?
I don't see why. That's a good question, actually. What's the format of the? Does Clemens happen to know the format of the images for codex? I think
yeah, but the end
right?
Yeah, by the end of the year there'll be QP TIFFs,
and that's perfect.
Yeah.
QP-TIFFs is more or less what I've been using with that works great in Visiopharm.
Okay, beautiful. The last question I have, unless someone throws on another question here, does the restaining and cover slipping alter the tissue and marker intensity?
That's a great question. Based on the paper that we took it from, it does not. If anything, they say it actually it might actually increase the sensitivity of the tissues. I will say that that particular paper is very interesting, and I had the pleasure of getting to see Dr. Cataretti talk in person, and he actually has a whole section in that paper for essentially keeping slides in a solution that maintains all the folding of essentially your proteins after antigen retrieval, and it's actually a trehalose-based solution. He really claims that after antigen retrieval, what you have is in the soup, right? The citrate soup that's in there is a lot of proteins. So it's kind of like a lot of dissociated proteins. So it's a little bit like when you have like Western running buffer, where you always add glycine to those running buffers. So he found that trehalose is actually equivalent to that, and he's shown that you can essentially keep a slide, decover slipped, keep a slide that already has undergone antigen retrieval in trehalose for months and months and months in the fridge, and then stain that again, and he doesn't see an appreciable decrease. He definitely does not see an appreciable decrease in the amount of signal on there. I will say that we've just begun using this, so I think that's a great control experiment, and it definitely should be done on a marker like cytokeratin or something where you know the signal's really good and you want to see it go up or down. But my guess is if anything, it would increase the sensitivity.
Great. Once again, thank you very much, Dr. Daniel. We really appreciate it.
Sure. Thank you very much.